So we are recording now. Thank you so much. Yes, the absent-minded professor here. Okay, so what we're going to do now, I'll talk a little bit about a few other hormones. We're going to describe the functions of thymosin and melatonin and their organs of secretion. We're going to define the placenta and list the hormones that it produces during pregnancy. And we're going to explain what happens in general to the endocrine system as humans age. Do you remember the pineal gland or the pineal body? It's part of the diencephalon, the epithalamus specifically. Well, it produces a hormone called melatonin, and that hormone helps establish our sleep and awake cycles. It may have some other functions that have yet to be substantiated. There is some research on some other functions of melatonin, but what we do know is that it sort of helps establish our sleep and awake cycles. For example, melatonin from the pineal gland does tend to secretion increases when we sleep at night, and we know that that if we were all to fly out to California, which is 2 hours behind us, tonight at 10 o'clock, it would feel like it's midnight because it would be midnight in Wisconsin. And that's because our bodies are sort of programmed to want to get up at a certain time and go to sleep at a certain time based on secretion of that melatonin. So melatonin from the pineal gland helps establish our sleep and awake cycles. The thymus is an organ that is posterior to the sternum or our breastbone. It's largest when we are infants and when we're children, and it becomes smaller and atrophies as we get older. I like to describe thymus as the place where our T cells go to college. Our T cells are very important immune cells, also referred to as T lymphocytes. They're produced in the bone marrow, and then they migrate to the thymus gland. And there they are taught to attack foreign cells and to leave self cells alone. So again, I like to say that the thymus is where T cells go to college. A very important hormone produced by the thymus is thymosin, and it's involved in educating those T cells to act appropriately and to attack and recognize foreign cells while holding harmless cells of the body, or what are called self cells or self antigens. There are many other tissues of the body that produce hormones, and some of these we'll talk about as we go through other organ systems like the cardiovascular system and the digestive system. But they're parts of the small intestine that produce hormones that you'll be introduced to when we cover the digestive system. The stomach also produces hormones. The kidneys, in the upcoming chapter, you're going to learn that the kidneys produce hormones that stimulate blood cell production. The heart produces hormones, and there are many other areas of the body that have endocrine cells and endocrine organs. So we talked about many major hormones that have major impacts on the body, but do understand that there are many other hormones that we did not talk about that are produced by other organs throughout the body. And this is something just to take note of. One hormone producing organ that I'd like to talk about is the placenta. The placenta forms during pregnancy. It's basically the junction that forms between the maternal circulation and the infant's circulation. So if we look on a pregnant mother, and if we were to magnify this area here, it looks something like this. Here you have the fetal circulation via the umbilical cord coming into close proximity with the maternal circulation. the two circulations do not actually mix, but they're very close to one another, and this allows for the exchange of nutrients from the maternal to the infant circulation, and waste products from the infant to the maternal circulation. Well, the placenta itself is a very important endocrine organ. It produces very important hormones, many of which are involved in maintaining this placenta, making sure it doesn't detach from the walls of the uterus. Some of those hormones include progesterone and estrogen. And there's this hormone that's produced very early on in pregnancy, within hours of fertilization, actually, called chorionic gonadotropin. Chorionic gonadotropin. That is actually the hormone that is typically used in early pregnancy tests. If you've ever seen or used the test where you take a stick and you place it in your urine stream, and if the little bar turns colors, it means you're pregnant. What that actually is doing is it's detecting the this chorionic gonadotropin in your urine, because it's released into the blood, and whatever gets into the blood typically finds its way into the urine. That HCG, what its function is to maintain the endometrium layer of the uterus to support implantation by the fertilized ovum, and then maintaining pregnancy. That, along with estrogen and progesterone, help maintain pregnancy. During pregnancy, the placenta grows, of course, in size. By the 3rd month of pregnancy, it actually produces more estrogen and progesterone than the ovaries themselves. So it's a major source of estrogen and progesterone during pregnancy. Estrogen and progesterone help maintain pregnancy, and they also help prepare the breasts for the production of milk, along with prolactin. Another hormone that I'll also bring to your attention, produced by the placenta, is a hormone, elastin. And as its name implies, it actually causes the pelvic ligaments and the pubic symphysis to relax and become more flexible in preparation for parturition, or giving birth. And as always, we're going to lastly talk about developmental aspects of the endocrine system. And as we talk about, aging, right, the information is typically never good, right? But you'll be happy to hear that for the most part, the endocrine system operates quite smoothly until old age. And if you're wondering what is considered quote unquote old age, over 70 historically anyway is considered old age. As we age, however, in general, there is a decrease or decline in the production of or secretion of hormones from our glands. That's what brings about menopause in females, as the ovaries produce less estrogen with age. Growth hormone production tends to decline with age as well. And overall, many endocrine organs decrease their output with age. Of course, much of this is also affected by diet and exercise. Exercise specifically has been shown to increase growth hormone production. And that is the end of our chapter discussion on the endocrine system, chapter 9. Let me make an announcement about the practical, and then I will open up the floor for questions before we move on to the next chapter. So Your practical will open tomorrow. It will open at 8 a.m. and it will close on Friday at midnight or 11:59 p.m. It's going to cover these labs shown here. These are all the labs that we have covered since the last practical. There will be 50 questions. You will have 42 minutes to answer those questions. The format's going to be pretty much the same as your lab quizzes. If you have accommodations where you get additional time, that additional time will be provided to you. I incorrectly said that you had to go through the testing center. I went back and read the communications that I received from the testing center and learned that was incorrect. I sent out an email, I believe it was yesterday, apologizing and informing you that I had told you incorrectly. So you will get the accommodations that you are entitled within this test in exam in Blackboard. A couple of questions I have received. Will there be a sheep brain dissection? Yes, yes, there will be. Will there be a dissected eye? Yes, there will be. Anything that is on your study guide is fair game, and anything that's on your study guide is reflected on the Quizlets. I'd never have you study something, waste your time studying something that I'm not going to then test you on. Now, it's not a huge percentage of the practical. I'm not going to have 10 questions from the eye or 10 questions anywhere near that number from those dissections, but they will be on the practical exam. Okay, so I'm getting a few questions here. Let me transition away from sharing my screen. Okay. What part of the gray mushy mass is the right kind of gray mushy mass? You need to clarify that a little bit for me, Emily, there. While Emily's clarifying that, any other questions? One thing I will say is I'm not trying to trick students. I'm not trying to make things any harder than they need to be. So rest assured that anything that I ask you to identify from the eye or from the sheep brain, they're going to be the more obvious, straightforward structures. Okay, well, let's go ahead and transition into chapter 10, which is on the blood. And the blood chapter is actually one of my favorite chapters. It's very straightforward. I think it's an easy read from your book. I think the chapter is only like 20 pages long. I think the material is straightforward. And honestly, everyone is at least somewhat familiar with blood. You've seen blood, you've touched blood, whether you like it or not, and you've given blood. And maybe even plasma, and you have some familiarity with blood. Now, I think that's always helpful when you're studying a subject, is to have some familiarity with it. So chapter 10 covers blood. We focus on the function and the composition of blood. Then we'll move on to chapter 11, which is the cardiovascular system. The cardiovascular system is the organ system that is responsible for circulating that blood throughout the body. So we first start with blood. What is it composed of? And then we move on to the cardiovascular system, where you'll learn how that blood is moved throughout the body, because the cardiovascular system's function is transportation. It uses blood as the vehicle to transport substances throughout the body. We're going to start by first explaining the function of blood, and then we're going to list the physical characteristics of whole blood. So, overall function of blood, it is transportation. It transports things. What kind of things does blood transport? It transports nutrients. It transports respiratory gases. We just covered the endocrine system, where hormones are secreted into the blood. So the blood transports hormones. It transports our immune cells. It transports waste products. And lastly, it helps distribute heat throughout the body. Some parts of the body produce a disproportionately high amount of heat, and the blood helps distribute that blood throughout the body. At the bottom of this slide, you'll see a statement that's taken directly from your book. Elaine Meribs wrote your author of your book, Blood is the River of Life that Surges Within Us. It was recognized hundreds of years ago that blood was necessary for life. People realized that if you lost too much blood, that would cause illness and possibly death. They didn't exactly understand how blood how it was responsible for maintaining life, but it was understood that it was necessary for life, and if you lost too much of it, you would die. Physical characteristics of blood. Well, of course, you know that blood is red, and we'll talk about why it's red later on today. Oxygen-rich blood tends to be a brighter, more brilliant, scarlet-type red. Oxygen-poor blood tends to have a duller, sometimes a grayish, bluish-like tone to it. The pH of blood stays in a relatively narrow range, 7.35 to 7.45, and blood is slightly higher than body temperature. Blood accounts for about 8% of your body weight. You have roughly 4.8 liters of blood in your body, and blood is the only fluid tissue. Does anyone remember of the 4 types of tissue, what type of tissue blood is? Is it nervous? No. Is it muscular? No. Is it epithelial? No. It is connective, right? It is connective tissue. You have a matrix, which is the plasma, and then you have cells dispersed throughout that matrix. Now that we've covered the physical characteristics of blood, let's move on to the composition of blood. So we're going to describe the composition and volume of whole blood, describe the composition of plasma, one of the main components of blood, and then we're going to look specifically at plasma, its functions, the 3 types of plasma proteins in plasma, and we're going to compare and contrast plasma and serum just so people understand the difference. All right, so let's say we were to take a syringe, and let's see here, who am I going to pick on? Let's say we went over to Michelle, and she's got good blood, and we took a syringe, and I withdrew some blood from it, and I used a heparinized needle, so that's an anticoagulant, so that the blood doesn't clot. And I then deposited that blood sample into a test tube, and then put that test tube into a centrifuge. A centrifuge spins sort of like your washing machine, and it uses centrifugal force to separate components in the test tube by density. The denser components fall to the bottom, the less dense components rise to the top. And so if I took a sample of whole blood from Michelle, put it in a test tube, and then placed it in a centrifuge and spun it, after a few minutes, I'd get a test tube that looks something like this. You'd see that there was a red layer at the bottom, an amber layer at the top, and this really little thin layer, white or sort of buffy coated layer here in the middle. And that would illustrate the 2 major components of whole blood. That amber liquid would be plasma, the liquid component of blood, and it makes up about 55% of the uh, volume of whole blood. The other 45% would be the formed elements. Those are the cells, the white blood cells, the red blood cells, as well as platelets, right? That the formed elements does include the white blood cells and platelets, but specifically they would be right there in that little buffy coat there. Almost all of the formed elements are red blood cells. Less than 1% of whole blood is white blood cells and platelets, which would be there in that buffy coat. Let's start with the plasma. The plasma is 55% of whole blood. The plasma itself is 90% water. So 90% of plasma is water. Well, if plasma is 90% water and whole blood is 55% plasma, what percentage of blood is water? Roughly 50%. Of that 10% of plasma, right, what makes up that 10% that's not water? Well, that's where the nutrients in your blood is transported. That's where your ions are found that are transported. Some of your respiratory gases are transported there. Hormones, waste products, and most of the solutes in plasma are actually your plasma proteins. Let's talk about those plasma proteins. So the most abundant solute in plasma, most of that 10%, are plasma proteins. And there are three plasma proteins you need to be familiar with. One is albumin. What does albumin do? Well, one, it acts as a buffer to regulate blood pH, and it also regulates osmotic pressure. It helps make sure that there's a certain amount of water that remains in the blood. Albumin is a charged protein, and so it attracts water into the blood vessels. Fibrinogen is a clotting protein. It is involved in the clotting process. You can't see it in blood unless the blood is in a process of clotting, and then it undergoes a conformational change, and then you see it. But it's involved in blood clotting. And then lastly are the globulins. There are two different types of globulins, what are called alpha and beta. You don't have to know it to that level, but some of the globulins are bound to lipids, and they help transport lipids through the plasma. Lipids are nonpolar, and therefore they don't dissolve in water, as you know. So if they're going to be transported through by blood in the plasma, they have to bind to globulins so that they can somewhat dissolve in the plasma. Other globulins act as antibodies fighting pathogens. So these are the three most abundant plasma. These are the three plasma proteins that you find in plasma. Often these plasma proteins are sampled in patients to make sure that those patients are healthy. In a healthy individual, these plasma proteins should remain in a very narrow range. As those plasma proteins are worn out or destroyed, the liver makes more. So if an individual were to have a low level of any of those plasma proteins, it might indicate some dysfunctions with the liver or some kind of disease or abnormality. Those plasma proteins are very important blood buffers. A blood buffer is a chemical that helps maintain pH. It resists changes in pH. We'll talk more about this when we get to the urinary system, but these plasma proteins help ensure that blood pH remains in that normal 7.35 to 7.45. When we get to the urinary system, you'll also learn that the lungs and the kidneys also play very important roles in maintaining blood pH. The plasma also helps distribute body heat evenly throughout the body. Remember that water acts as a heat sink. It's very good at absorbing heat, and so the plasma helps distribute heat throughout the body. Some parts of the body produce more heat than others. As blood flows, it helps distribute that heat evenly throughout the body. So sometimes your book, at least your book, used to sometimes talk about plasma, and then it would talk about serum, and it never really explained the difference. There's not a lot of difference, but I wanted to make sure that you understood that plasma and serum are not synonymous. They are similar, but they are not exactly the same. So earlier when I was saying I was taking a blood sample from Michelle, I was using an heparinized needle. Heparin is an anticoagulant, and I didn't want the blood to clot. So when I used an anticoagulant to take that sample and I spun it down in a centrifuge, I got plasma, and I got the formed elements here. Well, what if I hadn't used an anticoagulant and I allowed the blood to clot? When I spun that blood down, I'd get something that looked more like this. What you would see is a blood clot down here at the bottom, and then we would have above that clot serum rather than plasma. The difference between serum and plasma is that serum does not contain the clotting factors. The fibrinogen and other chemicals that are involved in a clot would be down here in the blood clot. Plasma contains fibrinogen and other chemical compounds and ions that would be in this blood clot. So sera and plasma are very similar, but they're not exactly the same. For the purposes of what we're going to be studying and learning about in chapter 10, it doesn't really matter. But that's the difference between plasma and serum. Just wanted to provide some clarification there in case you run into those words either while you're reading the book or in another class or even in the professions that you plan on pursuing. Okay, any questions about plasma before we move on to the formed elements? Questions about plasma? Okay, Lauren, what question do you have? You can text or you can unmute your mic and ask the question. Either one is fine. Okay, sure can. Well, it'd be nice if I share the slide with you. Sorry, let me go back and slide. So with plasma and serum, the only difference is serum does not contain the clotting factors. Clotting factors just all the substances that are involved in the production of a blood clot. Plasma can only be obtained if you use an anticoagulant when you take someone's blood. Otherwise, it'll clot. So where the anticoagulant prevents the clot from happening, and so you get plasma when you spin down a sample of blood. If you don't use an anticoagulant, what happens is just the turbulent flow of blood that just occurs by withdrawing that blood from the blood vessel will initiate the blood clot. And so the fibrinogen that's in the plasma, calcium, some other vitamin K, other compounds and ions that are in plasma that are involved in the clotting process, they'll all be down here in this blood clot. So the difference between serum and plasma is that serum does not contain the clotting factors plasma does. Okay, you are very welcome. So there was a question about different blood clots. Now I can't say definitively that different blood types won't have any impact whatsoever with the composition of plasma or serum, because certainly there might, but as you'll see on Wednesday when we talk specifically about blood types and blood typing, that mainly has to do with glycoproteins or antigens on the surface of red blood cells. Very, very welcome. Okay, let's move on to the formed elements. So the formed elements are basically the blood cells and platelets. So we're going to describe the composition of the formed elements. We're going to define hematopoiesis, make sure you understand what that is, and identify where it occurs. So we're moving from the plasma, we're now moving to the formed elements. So if the plasma represents 55% of whole blood, the formed elements represents the other 45%. And overwhelmingly, most of that is red blood cells, right? A very small component of that formed elements are white blood cells and platelets, okay? So erythrocytes are your red blood cells, and they make up of that 45%, they are 44+%. So 44+% of whole blood is the erythrocytes or red blood cells. Leukocytes, your white blood cells, and platelets, which are not cells, they're actually pieces of a cell called a megakaryocyte. We'll talk about later today or on Friday. The leukocytes and the platelets make up less than 1% by volume, and you saw this when we looked at the blood smears in the tissues lab earlier this semester. When you look under the microscope and you look at a blood smear, and that's just basically when you take a sample of blood, throw it on a slide, and put a slide cover over it. That's what you're looking at here. And most of these cells are, of course, red blood cells. You have a limited number of larger cells with these lobed nuclei. These are your white blood cells. And then we have these little dark staining specks. Those are your platelets. So what you saw in the microscope reflects the composition of blood. All this white space is plasma, making up 55% of blood, and then the other 45% of blood is mostly red blood cells with a small amount of white blood cells and platelets. Before we move on any further, let me mention something about the terminology leukocytes and white blood cells versus erythrocytes and red blood cells. Students always ask, do I have to know erythrocytes and leukocytes? Will you accept white blood cells, red blood cells? And the answer is yes. However, if the answer is red blood cells, can you write red blood cells? Yes. If the answer is white blood cells, can you write white blood cells? Yes. However, you are expected to recognize that erythrocytes are red blood cells and leukocytes are white blood cells, meaning that if I can have erythrocytes and leukocytes in a question, and if you were to ask me what do those words mean, I ain't telling you, because you are supposed to be able to recognize them. So while I'm not requiring you to answer erythrocytes and leukocytes, I may ask a question that includes those terms, and you need to know that erythrocytes are red blood cells and leukocytes are white blood cells. Hematopoiesis is blood cell formation, and it occurs in the red bone marrow of flat bones and the proximal epiphyses of the humerus and femur, at least in adults. In infants, it's pretty much in every bone, including the medullary cavities and such. All right, I saw that question, Emily. I'll answer that in just a second. All of the blood cells, the red blood cells, as well as the white blood cells, come from a common stem cell called the hemocytoblast. The reason why you constantly are having new blood cells produced is because there are these stem cells in the red bone marrow that receive different cues, hormones, tissue factors. They're all chemical signals that tell it to, hey, become a white blood cell, or become a red blood cell, or become a specific type of white blood cell. There was a question from, I think it was Emily, just asked a question about, well, what are lymphocytes versus leukocytes? A lymphocyte is nothing more than a specific type of white blood cell. There are different types of white blood cells. We'll talk about either later today or Friday, depending on where we get. Okay? At this point, I just want you to know hematopoiesis is blood cell formation. It occurs in the red bone marrow, and it occurs through the transformation or differentiation, I should say, of stem cells into various blood cells. The first form element we're going to talk about are the erythrocytes, the red blood cells. So we're going to describe the anatomy and physiology of erythrocytes. We're going to define anemia and list different sources. We're going to explain why erythrocytes have a relatively short lifespan. We're going to describe erythropoiesis, the production of red blood cells, and we're going to explain the homeostatic mechanism that controls erythropoiesis, or red blood cell formation. I really like talking about erythrocytes, or red blood cells, and the reason why I do is because their anatomy, their structure, is perfectly suited for a red blood cell's function. Red blood cells are these thin, disc-like cells. They're what's called biconcave. They are curved inward on both sides. So they don't have a lot of volume, but they have this enormous amount of surface area. And this is related directly to their functions. Their function is to carry oxygen from the lungs to the tissues and distribute that oxygen to the tissues. So they're thin, so the oxygen doesn't have that very far to diffuse, and they have this huge surface area that facilitates diffusion. If we then look internally at a red blood cell, you would see that red blood cells don't have a nucleus, and they have very few organelles. Instead, what you find is the cytoplasm is almost completely filled with a very important protein called hemoglobin, which we abbreviate HB. This is a hemoglobin molecule. Could you believe that I actually had to draw a structural representation of this in biochemistry? That was all kinds of fun. Hemoglobin, this is a hemoglobin molecule. The cytoplasm of the red blood cell is filled with hemoglobin, and each hemoglobin molecule contains a heme group. That heme group contains iron, and that in each heme group binds a molecule of oxygen. Each molecule of hemoglobin contains 1, 2, 3, 4 heme groups. So every molecule of hemoglobin binds to 4 molecules of oxygen. And in fact, this heme group, as I said, contains iron, and when oxygen binds to it, it forms iron oxide. Another name for iron oxide is rust. Rust, what color is rust? It's red. So when oxygen binds to hemoglobin, it causes the production or formation of iron oxide, which is rust, and that's why blood has that red color. And that's why oxygenated blood, that's blood that's high in oxygen, has a brilliant scarlet red color versus deoxygenated blood, which has a more duller bluish type color. These red blood cells are basically little bags of hemoglobin. So they're perfectly suited to their function of carrying oxygen. There are roughly 250 million molecules of hemoglobin in a red blood cell. If each hemoglobin molecule binds to 4 molecules of oxygen, how many molecules of oxygen roughly can each red blood cell hold? Well, when I was in school, 250 times 4, or 250 million times 4, was a billion. So each red blood cell can hold roughly a billion molecules of oxygen. So oxygen binds to hemoglobin inside a red blood cell. What this means is that the hemoglobin content of blood actually determines the oxygen carrying capacity of blood. So if you have a high red blood cell count, or if you have a large amount of hemoglobin per red blood cell, then your blood can actually carry a high amount of oxygen. And in fact, we actually see differences in red blood cell count between men and women. In general, men have 13 to 18 grams of hemoglobin per 100 milliliters of blood, and women 12 to 16. So men tend to have a higher oxygen carrying capacity compared to women. Of course, everyone is different and there is a range. I am not saying that every man has a higher oxygen carrying content capacity than every woman. That's not what I'm saying. I'm just saying these are averages. Certainly someone who engages in aerobic exercise that is female would be expected to have a higher oxygen carrying capacity than a male that is relatively sedentary. The point here is that it's the hemoglobin content of your blood that determines how much oxygen your blood can carry. As I mentioned, hemoglobin is an iron-containing protein. It binds strongly but reversibly to oxygen. And of course, we want it to be able to bind reversibly to oxygen because we want it to pick up oxygen in the lungs and then to release oxygen to the tissues. Anemia is a decrease in the oxygen carrying capacity of blood. Someone that is anemic can't carry the normal amount of oxygen in their blood. And there can be several reasons for this. One, what's called iron deficiency anemia. That is where there are normal amounts of iron in the blood just aren't, the normal amounts of iron in the blood just aren't there. It could be because the person has a poor iron diet. They just don't get very much iron in their diet. Or it could be because they've lost a lot of blood. A lot of times it's a little of both. We don't really lose iron by a significant amount of iron from our bodies except through hemorrhage. We'll lose a little in feces, but for the most part it's because of hemorrhage. Over the years, I've talked to many women who are vegetarians, and most of them eat fish because they tried being strict vegetarians where they didn't eat any meat at all. But the problem they ran into is they started becoming anemic, and the reason why is iron in plants is not as available. It's not as digestible and absorbable as iron in animals. And women lose blood every month through their menstrual cycle. So there's that increased iron loss in women versus men that increases their dietary need for iron. And so most people that I have talked to who are vegetarians that are women have had to at least eat fish. Not all, but most, because otherwise they became anemic. Without the iron, if iron is limiting, then it limits how much hemoglobin can be produced. Another cause of anemia can be a vitamin B12 or folate deficiency. Both of these vitamins are involved in the production of DNA. Well, how in the world would a deficiency in vitamin B12 and folate cause anemia? Well, red blood cells have to be produced all the time. We'll talk shortly about why, but they die, right? Every 120 days or so. And so new ones have to be produced. That requires mitosis, and mitosis requires replication of DNA, right? Mitosis and cytokinesis. If there's a deficiency of vitamin B12 and or folate, then that can limit how much DNA replication and mitosis can occur, and therefore you run into a situation where a person has a below normal number of red blood cells. So vitamin B12 and folate deficiency can lead to anemia. The last cause of anemia I'd like to talk about, and certainly this is not an exclusive list, is sickle cell anemia. It's a specific type of anemia due to a genetic disorder. Someone who has sickle cell anemia, when there is a drop in the oxygen content of their blood due to exercise or stress or anxiety, it will cause the red blood cells that are normally round to take on this sickle shape. And what that will cause is a decrease in flow of those sickled red blood cells. In fact, they'll dam up in certain parts of the vasculature of the body and deprive organs of oxygen. There may be problems getting these sickled cells to the lungs to pick up oxygen, and so there's a general decrease in the oxygen content of the blood. Now, something that I find fascinating that I want to share with you is the difference between a normal hemoglobin molecule and a sickle cell hemoglobin molecule is 1 out of 146 amino acids. There are 146 amino acids in a hemoglobin molecule. Someone with sickle cell anemia has hemoglobin where the 6th amino acid that's normally glutamic acid, has been replaced with valine. That's it. 1 amino acid out of 146 causes the cell to sickle when there's a drop in oxygen. Sickle cell anemia is, you find people carrying the it's a what's called a point mutation, where there's one mutation on a very specific location on the DNA molecule. That's what causes that replacement of that amino acid. You find it in people who are either living in the malaria belt of Africa, or they are descendants of people from the malaria belt of Africa. People who carry the trait for sickle cell, the sickle cell trait, that is, they had the gene for it but didn't have the disease, tended to have better survivability against malaria, before we had the healthcare that we have now. Okay, so we're talking hundreds of years ago. And so people had better survivability if they had that mutation. And so that caused that mutation to persist in populations from malaria belt or descendants thereof. What it allowed is their spleen to kill the plasmodium, which is the protozoan which causes malaria. Not something you have to know. It's just something it's appropriate to talk about it in the context of talking about anemia. Okay, I'm going to wrap up. Just give me one moment. We're going to wrap up our discussion on erythrocytes. What I wanted to mention was that in a adult erythrocyte, red blood cell, you don't have a nucleus. You have very few organelles. That maximizes the space that you have for hemoglobin and binding oxygen. But what's the trade-off? By not having a nucleus, not having ribosomes and endoplasmic reticulum, etc. Well, you're not going to be able to divide and grow and replace worn-out proteins, worn-out organelles. So erythrocytes have a relatively short life cycle. They die every 100, 120 days, right? They are broken down and phagocytized by the spleen or the liver, right? And that's why and they're replaced by erythropoiesis. Right? We're constantly producing new red blood cells because old red blood cells die out relatively quickly. Right? So we are at 12:23. I do not want to go over any further than that. We'll pick up from here on Friday. So this is the end of our formal portion of lecture. If you have any questions, please ask them now. I want to follow up on your question, Emily. So a lymphocyte is a leukocyte, but a leukocyte isn't necessarily a lymphocyte. That is correct. A lymphocyte is lymphocytes are basically your B and T cells, but those are not your only white blood cells. You have other white blood cells such as the basophils and the neutrophils and the eosinophils. Right? So like every orangutan is a monkey, but not every monkey is an orangutan. A lymphocyte is a leukocyte, but not every leukocyte is a lymphocyte.